technology. So I'm going to, pretty much what I'm doing, I'm recording the video and audio at the same time. So both of these things will show, audio on the blackboard and video on the YouTube, they will show right away. Like 15 minutes break, it's all there. Okay. Anyway, so what we covered so far? The cell wall synthesis inhibitors, right? Different types of penicillins, vancomycin, bacitracin, based on the different mechanisms. And we talked about the protein synthesis inhibitors, okay? And we talked about the drugs that inhibit, disrupt the membrane. And we figured that these guys, the polymyxins and daptomycin, they are really badass antibiotics, right? Because they can affect both bacterial membranes and eukaryotic membranes. That make sense? Just reminding you. So another process that all bacteria have is the DNA replication and RNA synthesis. Okay? So there's a drug, well, class of drugs called fluoroquinolones that pretty much block the replication of DNA. One of the representatives of this drug you definitely know, the ciprofloxacin, or we call it Cipro, yeah. Ciprofloxacin and other drugs from fluoroquinolone group, they bind to they bind to the enzyme that is called DNA gyrase. Synonym that you know is DNA topoisomerase. Does anyone remember what it does? Topoisomerase. Huh? It prevents supercoiling of DNA. When this enzyme doesn't work anymore, DNA becomes all tangled and replication cannot proceed. Does that make sense? So fluoroquinolones are the great drugs. They are broad spectrum drugs and effective against all kinds of bacteria. It makes a lot of sense because the DNA replication is something that can be found in all types of microbes. Okay, so by inhibiting this, we can affect pretty much everything. Now, another drug that I have a certain, not so much personal connection to, but I have special feeling to, is rifampin. It belongs to the class of rifamycins, and it inhibits the synthesis of RNA from the DNA matrix. So essentially it inhibits transcription. Does that make sense? It is distinguished drug because it's very active against Mycobacterium tuberculosis. It's a component of the modern combination therapy against that infection. My personal connection to it, I was studying bacterial transcription when I was a student, and rifampin was commonly used as just the inhibitor for the process in the research. Essentially, what it does, if you remember, in the process of transcription, DNA and RNA polymerase, they make a complex. RNA polymerase has a shape of like open fist. And then here, the RNA molecule is produced. Rifampin binds to the active site and prevents the release of RNA molecules. So there's no DNA and RNA polymerase can still make a complex, but RNA is not produced anymore. So this is essentially, since it's binding to the active site, it is a competitive inhibitor. Does it make sense? So these drugs are specific against bacteria. Why? Why they have high selective toxicity? Why they specific against bacteria and have fairly limited side effects? Because DNA replication and transcription in bacteria is different from eukaryotes. Does that make sense? Now, these drugs represent, remember when we were talking about the history, and I mentioned sulfonamide, the first truly synthetic drug was developed in Germany. So sulfa drugs are elaboration of sulfonamides. One of the critical pathways in bacterial cell is the synthesis of tetrahydrofolic acid. It starts with the fairly simple organic compound called para-aminobenzoic acid. The enzyme that is called dihydropteroate synthase converts it into the dihydrofolic acid. Dihydrofolic acid is then converted by the dihydrofolate reductase into the tetrahydrofolic acid. I hope with that monologue I scared the living daylights out of you. No, you don't have to know all the names. Really, you don't. You don't have to know the names of the enzymes, seriously. And you don't have to know the name of this or this intermediate. Although I expect you to know that the final purpose of the pathway is the folic acid synthesis, okay? And I expect you to know why it is important. Because folic acid is the precursor molecule for synthesis of nucleotides. Does that make sense so far? So sulfa drugs that are things like sulfamethoxazole, I bet you've heard about this, which is usually signed the acronym SMX. So sulfamethoxazole is the structural analog of para-aminobenzoic acid. Sulfamethoxazole binds to that first enzyme, to the active site of that first enzyme, and competitively inhibits it. Does that make sense? So it competes with para-aminobenzoic acid for the binding to the active site. By itself, sulfamethoxazole is bacteriostatic. Another drug from the same group that is called trimethoprim is the analog of the dihydrofolic acid and binds to the second enzyme, and also competitively inhibits the enzyme. Does that make sense? And by itself, trimethoprim is also bacteriostatic. When you combine them, combination of sulfamethoxazole and trimethoprim, often referred to as TMP SMX, is bactericidal. Right? That make sense? These drugs often are referred to as anti-metabolic drugs because they inhibit one of the critical metabolic pathways in the cell. Good? Is that understood? I'm going to ask you. Yes?
Can you restate the first part of it again? How it stops the nucleotides from being formed?
So, look, you have a pathway for eventually nucleotide. It's a synthesis of folic acid that is absolutely necessary for nucleotides, okay? So, two key enzymes in that pathway are dihydropteroate synthase and dihydrofolate reductase. That make sense? By inhibiting either of those enzymes, you can greatly reduce the amount of folic acid that is produced. By inhibiting both, by a combination of the drugs, you can pretty much completely stop the folic acid synthesis and the cell will die. Did I answer your question? No, the drug inhibits the enzyme. The drug this drug binds to the active site instead of para-aminobenzoic acid. Does that make sense? And this drug binds to the active site instead of dihydrofolic acid. Okay, so by themselves, individually, they are bacteriostatic, but when they are taken together, they're bactericidal. Right? There's something I wanted to mention, and I blank on something, completely blank on something right now. Oh, so pretty much you can say that these two drugs inhibit two different steps of the same metabolic pathway. Okay? Another anti-metabolite, sorry, is isoniazid, which is some azide of some nicotinic acid. Anyway, it inhibits synthesis of mycolic acid. And mycolic acid can be found in what kind of microbes? Mycobacterium. Yes. Mycobacterium tuberculosis, Mycobacterium leprae, Mycobacterium avium, and other mycobacteria. And although leprosy is not that big of a deal anymore, we have very effective drugs against it. It's fairly easily treatable. Mycobacterium tuberculosis is a big deal. And having isoniazid as a specific treatment for mycobacteria is also a big deal. Does that make sense? Okay. One of the recent metabolic inhibitors developed is diaryl quinolones. It's a whole class of the drugs. So what they do, they inhibit ATP synthase, essentially stopping oxidative respiration. Does that make sense? So remember, we have electron transport chain, and electron transport chain generates proton gradient, and then protons move through ATP synthase, and ATP synthase churns out ATP. Does that make sense? If you block it, you block the synthase, there's no ATP. And so, essentially dies. The problem with diaryl quinolones is that they're toxic. Like big time. They have pretty significant toxicity to the heart and to the liver. Okay, but you will see why we are somewhat desperate in terms of the drugs that we use now. Okay, because next, what we're going to talk about is resistance to drugs. Question, yes. Diaryl quinolones. Yes.
Those will also target eukaryotic cells. They're not
Yeah.
just for bacterial.
They will have,
So essentially all the cells are going to be shut down.
well, they more specific to bacterial electron transport chain, especially for mycobacteria. But they will have the same effect, not as pronounced, against eukaryotic, right? But if you even decrease ATP production in myocytes, heart is not going to beat as well as it should. And if you took ATP2, you know that you don't need much to induce various problems with the heartbeat, like arrhythmia, right? So you're still going to be screwed, okay? Maybe not immediately, but over time. And this is what the next topic is, resistance. We've already mentioned that Alexander Fleming, when he was delivering his Nobel Prize speech, he warned about development of resistance. And microbes inevitably do that. Now, how resistance occurs? What is... I expect certain answer from you, but I can't ask a proper question. So, what drives resistance? Mutations. Okay, great. Excellent answer. Mutations in what? In the DNA. So changes in the DNA, meaning changes in the genome, right? Now, why certain trait, like resistance, becomes seen? It can be seen in a population. Why certain trait actually happens? How to say it? What is the condition for evolution, actually? What is the condition for a certain trait to be passed through the generation? Huh? It should be reproducible, but since it's in the genes, it is reproducible. What else? What it should give the organism? What it should give? You narrowing it down, but what it should give to the organism?
Toxicity to it where it starts to get resistant.
What I'm saying? Huh? No. Just throw. What? Say. Why? Huh? Advantage. Advantage. Excellent. Advantage. You know what is the tallest nation in Europe? That's kind of a change of gears. What is the tallest nation in Europe? Danish. They have a tradition to marry really tall people. So they prefer tall men and tall women. Like, men prefer tall women and women prefer tall men. So it's a beneficial, it gives an advantage in mating. Does that make sense? That's why they selected, they kind of selected this trait. They told, okay? So in bacteria, unless the trait gives the entire population certain advantage, the trait is not going to persist. Does that make sense? So, if we would cultivate microbes on the regular TSA plate without any antibiotic, will there be any microbes with resistance? Yes. Will they take over the population? No, because there is no selective pressure and this resistance doesn't confer any advantage. Does that make sense? So the resistance is driven by the pretty much exposure to antibiotics. Now I'm going to ask you another question. This resistance, if we would go outside and isolate, hypothetically, a microbe from soil, could we find the resistance in that microbe? Microbe from soil, like in absolutely wild place, no humans there for hundreds of years. Yes, why? What is the original, But why it is there? Why this trait is beneficial for that microbe in soil? Huh? Why it is beneficial? Bingo, because half of the antibiotics that we use are naturally sourced. Originally, it's a chemical warfare. One microbes try to kill others. So there's a lot of intrinsic resistance. Does that make sense? There's a lot of microbes that are normally resistant to, say, penicillin or aminoglycosides like streptomycin, just because they have to, this trait gives them the advantage when they live in a community, in the close quarters with others. Does that make sense? Now, that is intrinsic resistance. And this, as Erin mentioned, is changes in DNA. So that's genetic, right? All these traits are in the genotype. You follow me so far? What microbes do with the genetic information? They share it. So therefore, resistance can be acquired by other microbes. Does that make sense? that is a very important thing to remember. That if we have, first of all, that resistance genes are somewhere there, right? They will be shared inevitably. And when we treat diseases with antibiotics, we apply that selective pressure that drives resistance faster. Let's put it this way. Because we throw so much pressure on the microbes that they will evolve much faster under those conditions. Does that make sense? So now I want you to play on the microbe side, on the bacterial side. So try to think like bacteria, and let's figure out how you can protect yourself against the drug. So you have a bacteria, and I mean, I'm fine, otherwise I wouldn't put this up, but bless you. So you have a bacteria, and you have a drug. You can look there, but it would be better if you would just try to think as a bacteria. How can you prevent drug from killing you? What are the options? Hypothetical options. Don't take it in. Okay. So you can block the penetration. That makes sense? What else? Huh? Counteract it. How? It's okay. Huh? Okay, break it down. Does that make sense? You break the drug in pieces. Okay? So inactivation. How else? Drugs. What do they do to microbes? For instance, penicillin. What does it do? But how? It stops the synthesis of the cell wall by doing what? It binds to something. To the enzyme. It binds to the enzyme. Okay? What if you slightly tweak the enzyme? Can you do that? Yeah, you can. I mean, as long as the enzyme still can do its job, but drug doesn't bind to it anymore, you're fine. So you can modify the drug target. Now, if you consume some nasty food or too much alcohol, how can you get rid of the toxins? You personally, as a human. You puke it out. That's what microbes do. They puke it out. They pump it out. Does that make sense? So those are pretty much like big major mechanisms. We're going to mention a few more. Okay? Now, what you have to understand, all of those mechanisms are mediated pretty much by some sort of a protein or change in the protein. So it means that resistance trait is because of the changes in the DNA. Does that make sense? So keep it in mind. All differences in phenotypes are reflected by differences in genotypes. So let's, yes. Yeah, go ahead.
DNA is only expressed because of environment. Could these bacteria already have the resistance that they didn't know unless the environment changes to where the DNA will express itself differently?
Terrific question. Yes. The short answer is yes. They may have a gene that they may be inactive in a certain environment, and we will discuss why it can be inactive. But when you expose microbes to antibiotic, this gene may become expressed. Yes. Yes, absolutely. It's absolutely possible. Now, modification. So, basic, yes.
I was going to ask if they get hide away from drugs too, as far as keeping resistance. You know, you say that microbacteria
Yes.
go into a thing.
Oh, it's not, it grows in the macrophage, yes. And it's probably, this strategy wasn't, so Mycobacterium tuberculosis and Mycobacterium leprae, they grow in the macrophages. I don't think this strategy was used to avoid drugs, it is definitely advantageous to avoid immune response, right? Because that's the last place your immune system will look for you. Okay. So mechanisms: modification of the drug, right? You can change the drug, like penicillinases. Remember, penicillinase is the enzyme, bacterial enzyme, that breaks down various types of penicillin, breaks down beta-lactam ring, right? So it modifies the drug, and drugs become ineffective. Okay? Is that understood? I will sum up what I expect you to know about the resistance. Don't worry. Right? You can prevent the uptake of the drug. Now, if the drug is big molecule, then it probably cannot move directly across the membrane. And then there's a cell wall that it has to crawl through. So there's a lot of obstacles. Now, there are proteins in the outer membrane of gram-negatives, inner membrane of both microbes, that are called porins. Porin. Which, what do you think porins do? Make a pore. They allow a passage, right? You alter, well, you as a microbe, alter the porin structure or decrease the number of the porin molecules, and drugs cannot effectively enter. Pseudomonas aeruginosa is the great example of multi-drug resistant microbe. It has so many mechanisms. It does so many fancy things. Okay, you can pump it out. Okay, now if you see the word, so pretty much pump the drug out of the cell. If you see the word pump, what type of transport is that? Active, which requires what? Keep it in mind that pumping the drug out requires ATP. Okay, so, well, let's just think about this. Drug will not kill the microbe, but there will be some fitness cost, because microbial cell will have to spend ATP for pumping the drug out. Does that make sense? And this will still confer some disadvantage. Modification of the target, that's what we were talking about when the enzyme is changed. For example, the enzyme that makes up the cell wall, the penicillin binding protein, right? That's the protein to which penicillin binds. Okay? So let's make a drawing, little drawing. Penicillin binds penicillin binding protein, PBP, and it gives, so, no cell wall. You follow me? It's like a generic scheme. Now if you change penicillin binding protein just a little, so drug doesn't bind anymore. Does that make sense? So I'm going to write no binding. Sorry, I didn't make a space between. Cell wall is going to be made now. Does that make sense? There are other ways to modify the target if the enzyme, if the drug binds to the enzyme, say drugs like chloramphenicol or aminoglycosides like streptomycin or tetracycline, they'll bind to different fragments of the ribosome. If the structure of the ribosome is slightly changed, just a little bit, just a small change, so that the drug doesn't bind, it doesn't inhibit anymore. Make sense? That's what microbes do. MRSA, for example, acquires the gene for the special form of penicillin binding protein, which says low affinity. What is affinity? What does it mean? How well it binds, yes. So low affinity penicillin binding protein will not bind penicillins really well. So there will be no inhibition of it. It will be still functional. Now these four are what I call elegant methods. Also, you can do not so elegant. You can simply overwhelm the drug with a target. Does that make sense? No? Say here, sulfonamides bind to block the enzymes. These two enzymes, right? This and this. If you make a shitload of enzymes, some of them are going to be inhibited, yes, but there will be plenty of molecules that are not. Make sense? That's very simple, not elegant at all. Okay? You just overproduce. Another mechanism is to take a detour, okay? Like over, do the bypass. In the winter, you have like a sidewalk. Try to block the sidewalk and people will start, walking around, okay? They're not just going to turn around, they're just going to walk around. So making a bypass, some microbes can do that. They may acquire mutations that will allow them to get around the roadblock that drug makes. For instance, effects of the drugs can be mitigated, like in case of vancomycin, okay? So when vancomycin will start binding to the outer layer of the cell wall rather than to the inner layer, okay, vancomycin will not be able to inhibit the cell wall synthesis as effective, okay? And finally, another elegant one. Now this one is elegant one. The target mimicry. How to explain this? I have a really bad analogy. Like the dog that humping somebody's leg, right? It humps thinking that it's another dog, but it's not. So that's what microbes do. They produce a molecule that looks like a target, but is not. So drugs bind to this target. Okay? Does that make sense? Yeah, they decoys. They produce decoys. So drug is sequestered by the decoy and the target is still functional. Does that make sense? Now, what I want you to know about mechanisms of resistance. Yes?
So, when you're talking about treating bacterial infection, bacterial infection that you're treating, is it mostly in the interstitial space or is it inside of infected eukaryotic cells?
Great question. I feel there's gonna be a second half of the question. I'm gonna answer the first. Depends on the microbe. Some bacteria, for instance, Staphylococci, Streptococci, E. coli, H. pylori, I think, they are mostly extracellular. Okay? Other microbes can be both. Trying to think of both, cannot think from top of my head. Mycobacterium technically can be both in macrophages and outside of macrophages. Some bacteria are strictly intracellular, like Chlamydia or rickettsia. So it depends on the microbe. Second part?
Okay, so then depending on where they belong, does it depend on which type of drugs are better at treating them?
Of course. Of course. Because some drugs penetrate better into the cell and some drugs penetrate worse. So it, of course, intracellular pathogens are generally harder to treat than pathogens outside, pathogens in extracellular fluid, right? Because if anything sits outside of the cell, even in the tissue, you can give a drug IV, it will migrate into the extracellular fluid and will reach the bacteria anyway. But if it's in the cell, you also have to think whether the cell itself, eukaryotic cell, is going to take drug in, how it's going to get there. So yes, absolutely, that definitely is an issue. Did I answer?
Yeah.
Excellent. So what I want you to know about mechanisms of resistance, what you have to kind of prepare yourself for. I'm going to give you a question that will describe a certain mechanism. For instance, the bacteria tra-la-la-la-la, okay, produces the enzyme that breaks penicillin. This type of resistance is modification of the target, which is no, modification of the drug, which is yes, target mimicry, which is no, overproduction of target, which is no. Does that make sense? So this may go either way. Either you have a statement, the description as a question, and then you have to pick a matching answer, matching name, or you have which of the following is example of target mimicry. Okay? Now with this, I will try to be as specific as possible. And during the exam, you're more than welcome, of course, to come to me and say, look, I'm not really sure. Do I understand it right? I will even make you a picture. Okay? Either for the statement or for each of the answers. Okay, so you can see what I'm asking. Because I understand sometimes it can be a little confusing. Is that clear? Okay. These are the guys that some of you have heard already. Some of you will inevitably hear. Those are major types, major classes of antibiotic-resistant microbes. So we're going to start with the most famous, MRSA. MRSA stands for, and that's another thing just up front, I expect you to know these acronyms. Okay? You may have a question, VRE stands for one of those, and you have to pick it up. That makes sense? Awesome. Because these are very strict ones. I mean, they have only one explanation. Methicillin-resistant Staph aureus. Remember the hype? And it's due. It's bad infection. I had a student who had a MRSA wound infection, and she showed me pictures. It was not looking good. I mean, she was cured by the time she was in the class, but she said it took her like 4 months to completely heal. And it was not a big one. It's just like this size wound in the knee. So it's pretty bad. So MRSA is resistant to Staphylococcus aureus, which is gram-positive microbe, right? It is resistant to beta-lactams, penicillins, methicillins, aztreonam, carbapenem, because it has so-called low-affinity penicillin binding protein. So those beta-lactams don't really inhibit. So it's a target that is modified. Make sense? We're going to put it here, target modification. And MRSA can be found mostly in hospitals, of course, but it is also in the community. Okay, it can be community acquired, and I have a wonderful sample. I can't reach it, but I will show you. We will do the gram-positive isolation maybe next week. I need to check the calendar. So one of the samples that I suggest you take is from your nose. And about quarter of the class, the number depends because the number of people is not very big, but about a quarter of the class is going to have Staph aureus in the nose. And if you want, we can check if it's resistant. We can put it on the plate and see if there's any resistance to beta-lactams. And it's out there. MRSA is out there. MRSA grows in people's noses, not causing anything. Yes?
MRSA the one that they have to actually like cut out? Which one is this? It's staph or MRSA that they actually have to cut?
Oh, it's both staph. It's Staph aureus, the Staphylococcus aureus. Cutting out sometimes is warranted if it starts to cause necrosis of the tissue. You can Google it yourself, necrotizing fasciitis. It's fascinating, pun intended. Really great pictures. We had a poster when I worked in the medical school. We had a poster of, patient with necrotizing fasciitis. So imagine something like not the size of that screen, but just slightly less, with all gigantic color pictures of rotting flesh. It's fantastic. It can cause pneumonia, and the worst thing, of course, is septicemia. Because if patient becomes septic, it's really hard to treat since it's so resistant. Another drug is vancomycin-resistant microbes. Vancomycin-resistant enterococci, VRE, and vancomycin-resistant Staph aureus. You may remember that vancomycin is another inhibitor of the cell wall synthesis. It works differently from beta-lactams. So it is often used to treat beta-lactam resistant gram-positive microbes. And inevitably, use of that drug caused the development of resistance in gram-positive enterococci and gram-positive Staph aureus. So these are nosocomial pathogens, especially enterococci. Enterococcus fecalis and Enterococcus faecium are normal components of human gut microbes. And people shed them, obviously, every time they take a dump. And when they take the drugs, those microbes develop resistance. It doesn't make them pathogenic immediately. I often say that many microbes are pathogenic only because they are put in the wrong place. VRE, vancomycin-resistant enterococcus, can cause urinary tract infections, wound infections, septicemia. So it means that it's not in the right place. It's not in the gut. It's not supposed to be in the wound. It's not supposed to be in the urethra. Does that make sense? And it's pretty hard to treat. Okay. Now this, we're getting to a couple of my favorite ones. So this is the extended spectrum beta-lactamase resistant gram-negatives. That's quite a treat just to say that. So these guys are still sensitive to carbapenems, which is good. Carbapenems are one of the last generations, belong to one of the last generations of antibiotics. Pretty much it's 5th generation as far as I remember. So what we say, gram-negatives, okay? E. coli, Salmonella, Shigella, Pseudomonas, all different gram-negative microbes that are resistant to drugs that contain beta-lactam, which means penicillins. Well, they're kind of lousy for gram-negatives anyway. Or 4th and 5th generation cephalosporins, which are broad spectrum. Okay? So a lot of those gram-negative microbes can still be found normally in the guts of people. So we talk about hospital-acquired infections. And along with that, those microbes often have plasmids that confer resistance to other antibiotics, like fluoroquinolones or aminoglycosides. So you see, it's often a combination. You may find yourself in a situation when patient has an infection resistant to multiple antibiotics. Okay? And these two are the feature of the day. CRE, carbapenem resistant Enterobacteriaceae. Enterobacteriaceae includes a number of bacteria that can be commonly found in the digestive tract of humans. Again, we're going to start with our favorite E. coli, Klebsiella, Enterobacter. Some people carry Salmonella asymptomatically. Can be even Vibrio, for God's sakes. Okay? So when these guys become resistant to carbapenem, they can cause hospital-acquired infection. Again, urinary tract or wound infections. Or pneumonia, like Klebsiella can cause hospital-acquired pneumonia. Now, why we pay so much attention to carbapenem resistance? I mentioned already that carbapenems were considered to be sort of a magic bullet, right? They're really good. They broad spectrum, can treat a number of infections, gram-positives, gram-negatives, and there's still little resistance. So about, I don't want to confuse, some time ago, in the last year, because I read it with my own eyes, in the last year there was a big news that carbapenem-resistant Klebsiella, which is one of the Enterobacteriaceae, appeared in India. It actually has a special enzyme that breaks down carbapenem, the carbapenemase, and the strain of that microbe actually got the name New Delhi strain. Bless you. The New Delhi is the capital of India. I have nothing against Indians. They're great people. They have lovely food. They have absolutely horrible movies. But there is a reason why many of resistant microbes come from India. Not from Africa, not from United States, not from South America. What do you think are the reasons? Huh? Come on, say it out loud. It's not a problem if it's not.
Talk about it, that they're not prescribing the right amount of antibiotics.
Yes.
Feel better.
Yes, lack of responsibility. Actually, from what I read, they have very loose regulations on who can prescribe antibiotics. Does that make sense? So it's not maybe a fully trained physician. It may be much less trained medical professional who can still prescribe antibiotics and treat people. So that really creates a bit of a problem. And this carbapenemase resistance is in the United States. It's problem number one. Second, it's on the plasmid. It's not chromosomal, it's on the plasmid, which means it's going to spread. Matter of time. It's real boned. Yeah, absolutely. I mean, it's not going to be explosive, but and I will give you an example. There was a paper published about 4 years ago about resistance spreading in developed country. Now this, so MDR, XDR, TDR stands for multi, drug resistant, extensively or extensive drug resistant, or totally drug resistant, Mycobacterium tuberculosis. Depends on how many drugs it can avoid. We talk about everything pretty much. Not only, so resistance to kanamycin or tetracycline is nothing new. But there are strains now that are resistant to linezolid, isoniazid, rifampin, drugs that used to be very effective against mycobacteria. And again, a lot of those strains come from India. But to be fair to my home country, we did put our share of efforts in this too. Because the TB problem back home is huge. Overpopulation of prisons. Not because we have so many criminals. Believe me, we're nice people. Just prisons are really small. Like really small. So they overpopulated. Infection spreads really fast. Treatment isn't perfect. So it's a lot of resistance comes from there as well. But Russian population of Russia is 140 million, which is like 7 to 8 times less than population of India. So our share is much smaller. There was a story about the guy actually who was diagnosed with totally drug-resistant tuberculosis here in the United States, and he wasn't legally banned from going anywhere, but he was strongly advised not to go anywhere. So he hopped on a plane and flew to his friend's wedding in India and then returned back. So they had to check 2 entire planes of people whether they acquired tuberculosis or not. Humans actually acquire it very easily, but only 5% of people who acquire TB ever develop the disease. Estimations suggest that about a third of the world population is actually infected. Not everybody's going to get sick. So for healthy, immunocompetent people, tuberculosis is a big deal, but we can deal with it, let's put it this way. But for immunocompromised patients, especially for HIV, it becomes a real hassle. And if they are infected with XDR or TDR strains, then treatment options are limited. Usually it's a combination of many drugs in the large doses over extended period of time. And those people aren't healthy in the first place. So for instance, in Africa, where percentage of HIV infected people in some countries, in some areas can be as much as like 25% of population are HIV positive. HIV and TB often go hand in hand. Okay, so that's a big problem. And we really have to be, again, mindful about this. So the last thing on the agenda for now is to discuss how we can assess the efficiency of antimicrobials. Well, first of all, we mentioned it last class. If you have a patient with like bleeding from eyes and nose, fever of 106, diarrhea and horrible rash, you're probably not going to test anything. You're going to give them broad spectrum antibiotics right now. But generally, it's not a bad idea to check which drugs microbe will respond to. Because you don't want to treat microbe that is resistant to penicillin with penicillin, right? That can be done using several ways. One that we used on Tuesday and which results we're going to discuss today is called Kirby-Bauer test. I want to remind you what you've done. So you have made a plate with the lawn, bacterial lawn. And then you applied different antibiotics. Okay, like on this picture, you have a comparison between Pseudomonas aeruginosa and Staphylococcus aureus. Each disc that you have applied is infused with a certain drug in the certain concentration. These concentrations are not taken out of nowhere, but they are relevant to clinically used drugs, clinically used concentrations. And then you look at the inhibition zone. Okay? For each microbe, actually, there is a strict criteria. each microbe is, say, hypothetically, Staph aureus. Those are not real numbers, just giving you, okay? So if the inhibition zone is less than I don't know, 10 millimeters, microbe is considered resistant. If it's more, it is considered sensitive. Does that make sense? So you measure it with like a ruler, and then you look at the table. Okay, of course, the larger the inhibition zone, the more sensitive microbe is. Is that clear? Now, what is the advantage of that method? Multiple antibiotics, one time. It's fast. It's like really fast and simple and fairly cheap. What is the disadvantage? Yes, it won't be able to distinguish between different types of administration, routes of administration. Can you change the drug concentration? No, you cannot. Here, you can. I mean, you have a you have like, I cannot see, of course, on this picture, nobody can, but say you have like 10 micrograms per mil. That's it. You don't know, what if you will go higher? What if you will go, can you go lower? Okay, so, but this method is often used to obtain so-called antibiogram. Have you heard of it? Hospitals, why there are hospital-acquired infections? Why? Why it's high there? Yes! Hospitals is where the sick people are. That's why you should avoid them, right? That's where the sick people are. And sick people, they have microbes, and those microbes they will rather infect sick people than healthy people. And it's a big problem because they're going to be on toilets, walls, floor, rails, whatever you name. Okay? So every so often, the clinical microbiologists in the hospital will go around the building and will sample different places. There's a SOP, I don't know if it's work instruction or SOP for that standard operating procedure. So in a certain way, in a standard way, they will sample, they will put it in a plate, they will isolate pure cultures, they will determine what microbe it is. And now it's fairly easy, okay? They probably will do gram stain, selective/differential media, maybe send it to the quick sequencing, stuff like that, okay? And then they will make a lawn like that, and they will determine what these microbes are sensitive to. Does that make sense? Because if you have a patient with nosocomial infection, you want to know which drugs to use, okay? And that's the regular procedure in antibiogram, right? Unfortunately, Kirby-Bauer test, as we mentioned, does not allow you to check many concentrations. You can get around this by using one of the dilution tests. So this is the old style dilution test. You see, the concentration of the drug decreases from right to left, okay? So you make dilutions of the drug. And at a certain point, it stops inhibiting the growth. So at 8 micrograms per mil here, the growth is inhibited, but at 4, it is not. So you can say that minimal inhibitory concentration is between 4 and 8. Does it make sense? Do you understand what minimal inhibitory concentration is? It's the lowest concentration of the drug that will inhibit bacterial growth. Another option is big 96 well plate. So you pretty much mix the microbe and the drug, but in very small amount, okay, and put it in those 96 wells. Then you have what a sterile control, the growth control, and you have different drugs. So for instance, clindamycin here, okay. So drug will not inhibit bacterial growth. All wells have growth. Does that make sense? Because this is how growth looks like. This is the growth control here. Okay. So it means that minimal inhibitory concentration is higher than whatever your highest is. In this case, it's going to be 32. Okay. Like penicillin, no growth here, so minimal inhibitory concentration will be 0.06. Now, of course, it is considered to be quantitative, but it's not 100% because you work with dilutions. You probably can you can make it more precise, but at the point of 0.06 micrograms per mil, it doesn't make any sense. You know, it's precise enough. The advantage of dilution test, minimal inhibitory concentration, and with this type, it's really high processivity. You can test many antibiotics in many concentrations, but again against a given microbe. Okay. Recently, another test, which is fairly convenient, was developed, so-called e-test. So in e-test, it's a type of dilution test to a certain degree. So you have a strip. It's picture number 4. This strip is infused with a drug. But this drug changes its concentration along the strip. Does that make sense? So it's highest here and lowest at the very bottom end. So you place it on the lawn, the plate, and you look at the inhibition zone. And you see that inhibition zone becomes smaller and smaller as the drug concentration decreases. The point where there is no inhibition is the minimal inhibitory concentration. So in this, it's going to be about 2. Is that clear? That's convenient and fast. Okay? Does that make sense? Now, say we are a pharmaceutical company who develops the new drug. And we want to know, and we have a few minutes to talk about actually drug testing. So what do we want to know about the drug? We want to know if it's what? Effective, and if it's safe. Okay, so we do safety and we measure the toxic dose. Unfortunately, we cannot do it in humans. Did I just say unfortunately? Yeah, well, unfortunately, but we can do it in humans. We can't test the actual toxic dose in humans. Sometimes it happens, but we can't, normally. We can test the effective dose. Let's say we are in the ideal situation when we can take a bunch of people and drug them, quite literally, and see in which dose people start to die of the toxic effects. Actually, not all 100%, but 50% of them. Okay, so the dose at which 50% of patients develop toxicity is called toxic TD50, toxic dose 50. Right? The dose which treats the disease in 50% of the patients is called effective dose 50. Does that make sense? Effective dose, you want it to be lower than toxic. Does it make sense? And the ratio between the toxic and effective dose is called therapeutic index. So let's say you have a drug which toxic dose is 200 and effective dose is 5. What is therapeutic index? 40. Therapeutic index should be higher. That makes sense? The higher the better. Because you have, why? Why it's better to have a higher therapeutic index? It doesn't necessarily work better, but why it is safer? I mean, it close to the toxic dose, closer, but my question is, you have a drug, you have two drugs, the effective dose is 1, whatever it is. Toxic dose for one drug is 100 and for another is 1,000. They're both going to be fairly safe. What you can do if the given dose doesn't work? Oh, you can crank it up, right? You can crank it up. And in the drug with a larger therapeutic index, you have more leeway to crank it up. Does that make sense? You have a better, like a more space to up the dose. Yes.
So do they, like, does the FDA, like, set a level that, of therapeutic index that it has to meet in order to be?
I don't think so. I don't know, but I don't think so, because there are many things that are taken into consideration when the drug is approved or not approved. For instance, the same, the platinum compounds for treatment of cancer. I think one of them called is cisplatin. They are extremely toxic, very toxic. But there are no good alternatives to them. So if toxicity is acceptable, if patients don't really die from the drug, then although they experience enormous toxicity, the drug will be approved. Okay, there are many points of consideration. There was a wonderful example of a drug with. I'm gonna explain you the situation, and you will tell me what was the toxic dose. So the drug was developed to treat something in horses, some kind of infection, and the experimental animal, the horse, was treated with the drug, and it worked wonders. Pretty much cured the infection in half a day. The horse was given a drug in the morning, and by the evening, the horse was fine. And next morning, the horse was dead. What's the therapeutic index? It's 1. So toxic dose and effective dose were equal. Well, I mean, it was experiment, but that's example of a drug with a toxic dose 1. Now, I'm going to ask you a question. What is the problem with testing antibiotics now? No. I mean, I'm not asking, we understand the resistance, yes. What I'm saying is we have a drug, like we have a prospective new antibiotic, right? We don't know how it's going to work in people. We have some data in mice and monkeys and whatnot, but we need to put it, and we've done safety study, so we know that it's not toxic. How can we do the efficacy study? How we technically should test every drug? Double blind, placebo controlled. Can we do it with antibiotic? Yes, we cannot. We can't take people, I don't know, with syphilis and prescribe half of them placebo. So how would you do that? You can, but then you're risking not treating them. They healthy. They're not sick, yes. Yes. You supplement the old form of treatment. And in different forms, you compare the new one to the old one. So often it is done like you have an old antibiotic, and one group of people is on the old treatment and another on the old and new. And if old and new is better, then you may start excluding the old and comparing the new and the old. And if new is better than the old, then it's good. It's sometimes called inferiority study. So it's really not that easy, double-blind, placebo-controlled. Okay, let's take a break now.